Hi guys, this is Dr. Lorenz. Today we're going to be covering different neurotransmitter systems, including the central activating systems, as well as how neurotransmitter systems adapt to repeated exposures to stimuli. So as a little caveat before we talk about how different neurotransmitters, individual neurotransmitters, interact with large activating systems, it's important to recognize the fact that the association between neurotransmitters and behavior is really complex. There can be a single neuron that uses in different neurotransmitters to talk to different of its neighbors. So it might use one neurotransmitter to talk to its neighbor on this side, but a different neurotransmitter to talk to its neighbor on that side, which means that at any individual synapse, right, the space between the neurons, there may be multiple different neurotransmitters acting. And the effect of this blue neurotransmitter might depend on the presence or absence of this purple neurotransmitter here. So each individual neuron can have different receptor types. It can be responsive to different kinds of neurotransmitters. An individual neurotransmitter can have a different effect depending on what other neurotransmitters are around. The neurotransmitter can have very different effects on a postsynaptic receptor, right, when it's a message that's being received by another neuron, versus its effect on the autoreceptor, on the presynaptic presynaptic membrane. And it can depend on where within the nervous system we're talking about, right? There are some neurotransmitters that will have an excitatory effect at certain parts of the body or certain parts of the brain, and an inhibitory effect at other parts of the body. So there's no such thing as a direct one-to-one relationship between an individual neurotransmitter and its effect on behavior. It's far too simplistic to say things like, oh, serotonin is the happy neurotransmitter, oxytocin is the cuddle hormone, right? Because these neurotransmitters will have radically different effects depending on the context in which they're working. So all of that said, all of that being kept in the back of your mind as we move on, there are a few generalities that can help us understand the actions of different neurotransmitters within their most prevalent systems. So starting with the somatic nervous system. The somatic nervous system is going to use predominantly acetylcholine. So acetylcholine's action is to stimulate skeletal muscles, that is our voluntary muscles, to cause contractions of those muscles. And an important receptor type for acetylcholine is actually the nicotinergic acetylcholine receptor. So nicotine is a drug that we take into our bodies, but the reason why it works is because it acts on these acetylcholine receptors that will become activated when nicotine lands on them. So when acetylcholine or nicotine binds to this receptor, the pore of this membrane will open up to allow ion flow, which will depolarize the muscle fiber directly. I always find it kind of interesting that nicotine activates the somatic nervous system and actually stimulates the muscles, because many people say that they smoke, they use nicotine to relax. But if you actually bring them into the lab and you measure the degree of muscle tension in the lab, their muscles are actually much more active and they are much more physically tense. So you have to think that nicotine's relaxing effects could be due to acetylcholine's effects on other parts of the brain, or it could just be psychosocial or psychological effects of taking a break from your routine to go have a cigarette, right? To have the social aspects of smoking and so on. Also, your public service announcement for the day. One of the ways that smoking can really impair athletic performance is through nicotine's actions on acetylcholine, right? So not only does smoking diminish your lung capacity, but it also really messes with the muscle's ability to become stimulated by the low levels of acetylcholine that are typically generated during individual muscle contractions, because nicotine usually we take a lot of nicotine in, and it can actually over time very subtly impair the way that the muscles are able to respond to messages from the brain to activate. So this is not really going to be a factor for everyday activities, right? Smokers can still walk around, but for really elite athletes who are right on that edge of trying to, shave off 1 second from their athletic performance, it really can impact the degree to which the muscles are able to respond to those split-second decisions. So your public service announcement for the day is, if you are an athlete or a dancer or someone who really relies on your physical performance, smoking is not a great idea. I have a little demo for you here to further illustrate the fact that the somatic nervous system uses acetylcholine. So you may have noticed that I said that acetylcholine will stimulate the muscles, right? But how do the muscles relax? Well, the muscles relax when there is absence of acetylcholine stimulating them. Muscles are naturally relaxed at baseline, at rest, and they need to have acetylcholine activating them to become tense. Now, there are some times when we are just really physically tense, and this tends to come up when we're really anxious or stressed out. We'll carry tension around, not even knowing that we're doing that. And so there's a technique that you can do to help relieve some of that underlying physical tension by temporarily exhausting the muscle's supply of acetylcholine, or rather the neuron that is stimulating that muscle. And we call this progressive muscle relaxation. This is something that I do with my patients who are very physically tense when they're anxious. So we're going to try this together for just a second. Take a moment to pull back from your computer and close your eyes. And you're going to concentrate on the muscles around your eyes. So you're going to spend the next 10 to 15 seconds or so tensing up the muscles around your eyes. You're going to try and squeeze your eyes shut as tight as you possibly can. Try to imagine all of the muscles in your upper face are trying to just squeeze into your eyeballs. So you're wrinkling your brow really tight. You're tensing up your forehead. You're going to squeeze your temples in. You're going to pinch your nose up as tight as you can, tight, 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 tight. Really keep it tight. And eventually it's going to start to feel really warm, and just keep going and keep it tight as you possibly can. Tight, 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 tight. And we're going to try to exhaust the muscle. So you're going to have to keep it, even when you're starting to feel like, oh my God, it's actually starting to get really warm, almost to the point of burning. And now let it go. Just relax all of the muscles in your face. Take a second to smooth out the muscles in your forehead and in your eyebrows. Relax your eyes. Let your eyes kind of gently relax. Let the temples pull back. Let your nose come down. Stop wrinkling, and let your cheeks become relaxed. And you'll notice, if you do this a couple of times in a row, that those muscles, you may not have even realized that they were tense before. But when you do this enough times, it exhausts the supply of acetylcholine to those muscles. And the muscles are able to become far more relaxed than if you just took a second to try and relax them without depleting the supply of acetylcholine. And it's amazing what this can do to our sense of anxiety when we tell our body to physically relax. It actually can cause some mental relaxation as well. So hopefully this is a useful trick for you, but if nothing else, it'll help you to remember that the somatic nervous system, the voluntary muscles, use acetylcholine as their primary neurotransmitter, and that relaxation of the muscles occurs when there's no more acetylcholine left to stimulate those muscles. All right, moving right along. The autonomic nervous system, which consists, as you may recall, of the sympathetic and the parasympathetic nervous systems, these are going to use the acetylcholine and norepinephrine as their main neurotransmitters. So the sympathetic nervous system, your fight or flight response, will predominantly use norepinephrine to talk directly with the organs that are going to become stimulated to elicit that fight or flight response. And the parasympathetic nervous system will use acetylcholine to stimulate the muscles to relax. Excuse me. However, it's important to know that when it comes to the blood vessels, both the sympathetic and the parasympathetic nervous system are going to activate different responses within the blood vessels to either increase blood flow or decrease blood flow, increase blood pressure or decrease blood pressure. However, the blood vessels will also use nitric oxide to help do some of the fine tuning at the capillary level. So nitric oxide will communicate with the sympathetic and parasympathetic nervous system to help with that fine tuning. The enteric nervous system uses a wide variety of neurotransmitters. So in addition to some of the neurotransmitters that are used outside of the enteric nervous system, such as histamine or dopamine and nitric oxide, there are also some neurotransmitters that are found either predominantly or exclusively in the enteric nervous system. So things like the pancreatic polypeptide, secretin, and so on. And these are going to help the regulation of digestion as well as metabolic processes and so on. And as well, your gut bacteria will produce certain neurotransmitters that your gut will then use to help coordinate its own digestion. There's actually more bacterial cells living in your gut than there are your own cells in the rest of your body. So if you think about it, there's actually more bacteria in you than there is you. And all of these gut bacteria can potentially contribute to the neurotransmitters that are used within the enteric nervous system. So we've talked a little bit about the somatic nervous system, the autonomic nervous system, and the enteric nervous system, so all parts of the peripheral nervous system and the different neurotransmitters that they use. So now let's switch gears and talk about the central nervous system and the neurotransmitters that are used within the central nervous system. So there are dozens of different neurotransmitters that are used within the central nervous system, but there are 4 really important systems that we call the 4 activating systems. So these are the neural pathways that work together to produce particular complex functions. So these activating systems are going to have cell bodies that are located in some nucleus within the brainstem, which is then going to send their messages via axons distributed throughout a wide region of the brain. So the 4 major systems that we're going to be talking about, the 4 major activating systems in the central nervous system, are going to include the cholinergic system, which uses acetylcholine, the dopaminergic system, which uses dopamine, the adrenergic or noradrenergic system, which uses norepinephrine or noradrenaline, and the serotonergic system, which uses serotonin. So all of these different systems are going to have certain features that are going to be relatively exclusive to that neurotransmitter, but they're all going to have some overlap with other systems. So when you think about a really broad construct like mood or cognitive function and thinking ability, right? All of the different neurotransmitters are going to play a role in that broad concept. But when we kind of dig down a little bit deeper and we look at things like, for example, reward and motivation, well, dopamine is going to play a really, really big role in that. Serotonin, norepinephrine, and acetylcholine are going to contribute to elements of that, but dopamine is going to be the kind of star player there. So let's go through each of these. And each of these diagrams that I'm about to present are all in your textbook, so don't try and write down every single word from these slides. Really try to cue into the overall here. So the cholinergic system is going to use acetylcholine as its primary neurotransmitter. So its beginning, its nuclei, the midbrain nuclei, are going to project up into the basal forebrain nuclei, and from there they will have very wide connections throughout the entirety of the cerebral cortex. The acetylcholinergic system is incredibly important for maintaining attention and alertness, particularly in wakefulness. It's also thought that it contributes to memory by helping to maintain neurons' excitability, which means essentially that when you are forming a memory, your neurons need to be a little bit more active in order to help lay down the connections between neurons. That is what constitutes a memory. And so when there is sufficient acetylcholine in the cholinergic system, it helps to keep these neurons alert and activated while they are forming memories. And when there is the slow death of acetylcholinergic neurons, this can contribute to elements of memory loss that are very common in certain kinds of dementia, such as Alzheimer's disease. A less extreme version of the forgetfulness that we see due to the loss of the cholinergic system is the loss of, or the forgetfulness and the distractibility that we see in very severe depression. So in folks with very severe mood disorders, in particular depression, we'll see a lower activation of the cholinergic system, which contributes to the sort of mental fuzziness that comes along with very severe depression. The dopaminergic system is going to use dopamine as its primary neurotransmitter, and there's two major projections within the dopaminergic system. They're both going to start in the midbrain, but they're going to go to different parts. So the nigrostriatal pathways, the orange pathways here, are going to start in the substantia nigra and will project up into the caudate nucleus, sort of in the midbrain here. The nigrostriatal pathways are incredibly important for maintaining normal motor behavior, particularly in the voluntary movements. These pathways will degenerate in Parkinson's disease, and so this will contribute to the muscle rigidity and the inhibition of motor free-moving motor commands, smooth motor behavior. The smoothness of motions are inhibited in Parkinson's largely because of the loss of the dopaminergic activity of this pathway, of the nigrostriatal pathway. This is one of those instances where I wish I could actually just show you guys what it looks like. Think of a jerky movement as opposed to a smooth movement, and you'll kind of have an idea of what it would look like when these pathways are disrupted. The second main dopaminergic pathway are the mesolimbic pathways. So the mesolimbic pathways are going to start in the ventral tegmentum, or the ventral tegmental area, and these are going to project upwards through the front part of the brain into the nucleus accumbens of the basal ganglia, as well as up into the prefrontal cortex. And these are going to be the major reward pathways. They are going to be what stimulates the feeling of reward associated with motivated action, and essentially your sense of pleasure, your sense of interest comes largely from activity of this mesolimbic pathway. So understandably, this is the pathway that tends to be affected by many addictive drugs and behavioral addictions, so things like gambling or compulsive shopping, those sorts of things. It's also thought that increases in dopaminergic activity within this pathway can be associated with some of the hallucinogenic and psychotic components of schizophrenia. So schizophrenia is actually a very complex disease. It has both mood-related components and social components, as well as hallucinations and disruptions of thought processes. So it's more the latter two, the disruption of thought processes and hallucinations, that are associated with higher levels of activity of this mesolimbic pathway. There are other things that are going to contribute to schizophrenia's effects on some of the other symptoms of schizophrenia. But it's kind of interesting that dopamine plays a major role in motor behavior as well as in reward behavior. And kind of an interesting feature of that sort of dual relationship is that medications that we give to try to address, for example, the muscle issues associated with Parkinson's, their side effects, if you give a level that is too high for that individual, the side effects may end up actually looking very much like schizophrenia. So it's very careful not to give too big a dose of the Parkinsonian medications because it can actually result in generation of hallucinations. And similarly, folks who take medication to help regulate their schizophrenia, in particular their psychosis and hallucinations, if they take those medications for a really long time or they take really, really high doses of those medications, it can actually result in some of the muscle twitches, and muscle rigidity, very similar to that which we see in Parkinson's disease. And so there's this sort of directional relationship between these two systems that can be impacted by the medications we give to treat one or the other side. So here I'll just mention again, because one of my research interests is in depression, one of the things that depression is thought to do is impair the ability of the mesolimbic pathway to stimulate reward when somebody has done something that is typically rewarding for them. Okay, the noradrenergic system, also sometimes called the norepinephrinergic system, is going to use noradrenaline or norepinephrine as its primary neurotransmitter. By the way, in this case, noradrenaline and norepinephrine are essentially two different ways of saying the same thing. So the noradrenergic system is going to start in the locus coeruleus within the midbrain, and is going to project up through the thalamus and then into many, many sections within the cerebral cortex, including into the cerebellum. The noradrenergic system is thought to be important for learning by stimulating a change in the structure of a neuron in response to experience. So if you remember when we talked about this at the very beginning of class, if you learn something, what that means is that your nervous system has literally changed its structure in some way. And so one of the ways that it can do that is by changing the structure of individual neurons, and it's thought that the noradrenergic system, and in particular the activity of norepinephrine, is going to contribute to those changes. It also will help to facilitate the normal development of the brain by essentially directing certain aspects of development, directing certain neurons to grow in one direction, not another direction, and also in organizing movements. The noradrenergic system is also important in activating or in maintaining kind of a stable emotional tone. When we say emotional tone, we mean that this person is neither too high or too low. So helping to maintain balance, emotional balance. So when folks have significant increases or decreases in activity of this system, it can lead to an imbalance in particular mood types. So decreases in activity of this system are thought to be related to the slowing and the sort of down part of the feelings of depression. And significant increases in activity of this system are thought to be related to the manic aspects of bipolar disorder, so the overexcited behavior. The overactivity of this system can also lead to the impulsiveness associated with suicidal behavior as well as parasuicidal behavior. So that is behavior that is intended to be self-harming but not intended to kill this person. So it's important to note when we're talking about increases here, decreases here, we're not talking about increase in the amount of norepinephrine or decrease in the amount of norepinephrine. So it's not like I could go into the brain and I could take a little sample of the amount of norepinephrine that's in the brain and say, okay, well, this person's likely going to have depression or this person's likely going to have mania. But rather, it's a decrease in the activity of this system that tends to use norepinephrine as its main neurotransmitter. So that could happen in a couple of different ways, right? That could happen because there is a decrease or increase in the overall level of that neurotransmitter, but it could also happen because the systems that are interacting with these neurons are either stimulating it or not stimulating it. So it's important, again, to remind yourself that it's not, there's no direct one-to-one. So even when we're saying an increase in norepinephrine is thought to be related to mania, that's not exactly correct. It's an increase in activity of the norepinephrinergic system that is thought to be related to mania. And then the last of our 4 central activating systems is the serotonergic system. Here we use the abbreviation 5-HT, which is short form for 5-hydroxytryptophan, which is the chemical name of serotonin. So the serotonergic system is going to start with the Raphe nuclei in the brainstem and in the midbrain, and as you can see, it goes all over the brain, right? It goes crazy Q all over. So you can probably get the idea that serotonin is used in a wide variety of cognitive functions. So serotonin is incredibly important for maintaining wakefulness. It's also important in helping to contribute to learning, particularly in mood-related learning. It contributes to focus and attention. So disruptions of serotonin can lead to obsessiveness and rumination, which can lead to obsessive compulsive disorder. It can also lead to the rumination that's very common in anxiety, worry, as well as in depression. It imbalances in serotonergic systems can lead to depression, can lead to sleep disorders because of its importance in wakefulness. So it can lead to either insomnia or, in more severe forms, narcolepsy and sleep apnea as well. So it's also related to disruptions of executive functioning and decision making. So you can see that a disruption of the serotonergic system can have a wide range of different effects. So as we've gone through, I've talked about how various different systems are related to depression. Depression's a really complex disease because it has a lot of different symptoms associated with it. And no two people who have depression are exactly alike. You can have somebody with depression who has really sad mood and who sleeps a lot and eats too much, but you can also have someone with depression who is really physically agitated, irritable, has trouble sleeping, has trouble making decisions, is forgetful, but has no particular sad mood, isn't crying or anything like that. So the diversity of different symptoms in depression kind of suggests something about its neurologic origin. So take a moment in your notes to write down what you think this might be. So the correct answer here is both B and C. Multiple different neurotransmitter systems are changed by depression. So we don't actually know what starts first, if depression changes neurotransmitter systems or if the change in neurotransmitters leads to depression. But the reality is that almost every neurotransmitter system is going to be changed by depression in some way. And so probably there's no one system that's the sole cause. And because multiple different neurotransmitter systems are impacted by depression, there are many different kinds of drugs that could potentially impact different neurotransmitter systems, which could help different people with different symptoms of depression, which is why it can take some time to cycle through a couple of different medications to figure out which one is going to be the right one for each individual patient. I mention all of this because your book strongly implies that depression is simply an issue of serotonin, and in fact strongly implies that it's just low serotonin. And that's a really misguided way of thinking about it, because in reality, all of the neurotransmitter systems that we've covered so far are going to be impacted by depression, and there's going to be symptoms of depression that are going to be caused by imbalances within each of those neurotransmitter systems. In this last part of the lecture, we're going to switch gears and go from talking about individual neurotransmitter systems and start talking about how different neurotransmitter systems might respond to repeated exposures to the same stimulus. There's 2 major ways that a synapse can change in response to repeated exposures to the same stimulus. So one way is habituation. With habituation, you'll see a decreased behavioral response to a stimulus over time with repeated exposures to that same stimulus. So think of when you put your shoes on this morning, right? You might have felt your shoes on your feet the first second that you put them on, but very soon thereafter, with the same stimulus coming in from the sensory neurons in your feet over and over and over again, over time you kind of noticed it less and less. And that's your feet habituating, the sensory neurons in your feet habituating to the sensation, this repeated exposure to the stimulus of shoes, 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 shoes. So habituation at a neuronal level occurs when the calcium voltage-gated channels at the very tip end of an axon, in the axon terminal, when these calcium channels decrease their sensitivity in response to an action potential. So with the same response coming in over and over and over again, not carrying necessarily any new information, these calcium channels over time will eventually become less and less sensitive to the message that's coming in from the action potential. So even though an action potential is coming in, coming in, coming in, the calcium channels are becoming less and less responsive to that signal. And essentially their threshold is becoming more variable. So these calcium channels are less and less likely to open up in response to an action potential. So when that happens, less calcium can come into the terminal bud and bind to the vesicles in the presynaptic membrane. So fewer vesicles will be pushed towards the end of the axon, and fewer vesicles will dump their neurotransmitter contents out into the synapse. What that means is there will be less neurotransmitter available in the synapse to stimulate the postsynaptic receptors. So there'll be less depolarization of the postsynaptic membrane. So the postsynaptic cell is going to be less likely to be stimulated to fire. So over time, what this will look like is even though this neuron, the presynaptic neuron, is being stimulated by an action potential, it's less likely to stimulate its neighbor neurons into firing because less neurotransmitter is being released. This can occur in either ionotropic or metabotropic receptors. So across either ionotropic or metabotropic receptors on the postsynaptic membrane. So think a moment about why this might be, evolutionarily speaking, why would this be adaptive for neurons to reduce their sensitivity to repeated information from the same source? Take a moment to think about what your answer might be. So the answer here would be C. It downplays information that doesn't change, which allows new information to be highlighted, right? So particularly at the level of your consciousness, the activity of your central nervous system, you need to really pay the most attention to information that's changing. Information that's not changing most of the time is not as important. And so if a neuron is getting repeatedly stimulated with exactly the same stimulus over and over again, once it's gotten that message through to its neighbor neurons, it doesn't need to continue to send that message at the same frequency. And so the neurons will become habituated. So they'll have less of a behavioral response to repeated information from the same source. The opposite of habituation is sensitization. This is far more rare. Sensitization is when the behavioral response to a stimulus increases with repeated exposures to the same stimulus. So at both the behavioral and molecular levels, habituation and sensitization are opposites. So in sensitization, when a neuron receives the same stimulus over and over again, it will actually release more neurotransmitter and will make the next neuron significantly more likely to fire. So the way that it works in sensitization is that there'll be some interneuron, so some third party here, that will release serotonin onto some of the presynaptic neurons' axon receptors, or receptors that are embedded within the membrane of the axon. And so these serotonergic receptors will actually activate potassium channels, which will prolong the effect of an action potential when it comes through. So when an action potential comes through, it'll last longer, essentially, and it will keep those calcium channels in the action terminal open for longer. And what that does is it will push more of these vesicles towards the synapse, and so more of these vesicles will exocytote their neurotransmitter into the synapse, and more neurotransmitter will be released onto this postsynaptic cell, so make it more likely to fire. The sensitization response requires there to be receptors with second messengers, right? The serotonergic system is a very important aspect of the sensitization response. And so it relies on metabotropic receptors. You have to have metabotropic receptors to have sensitization occur. This kind of response tends to occur for emotional stimuli. So for example, PTSD is a really kind of key example of this. So when there is some sort of traumatic stimuli, something that reminds the system of a trauma that it's experienced, it will actually increase its behavioral response to stimuli that look like that stimuli that previously caused some sort of traumatic event. And so over time, it'll actually significantly increase its response with repeated exposures to the same stimulus. So just as review, habituation versus sensitization. Habituation is going to decrease the amount of calcium that flows into the presynaptic neuron, which will decrease the amount of neurotransmitter that is released into the synapse. And because you'll have less activation of the postsynaptic neuron or postsynaptic cell in response to activity of the presynaptic cell, it'll over time weaken the association between these two neurons. So when one neuron fires, the next neuron is actually less likely to fire. So it's weakening the association between these two. In contrast, sensitization is going to increase the amount of calcium that comes into the presynaptic neuron by prolonging the effects of the action potential, which in turn will increase the amount of neurotransmitter that is released into the synapse, which will make it more likely that the next neuron or the next cell is going to fire. So it will increase the association between two neurons, because when one neuron fires, it'll make it more likely that the next neuron or the next cell will become activated. Habituation can take place in either the short term or the long term. It can take place over a few seconds, or it can take place over minutes, hours, days, sometimes even over a lifetime, depending on how strongly this response is occurring. However, sensitization is typically only going to have short-term effects, just a few minutes at most. Habituation can be present across all different kinds of stimuli, sensory stimuli, emotional stimuli, whereas sensitization is mostly only going to occur for emotional stimuli, such as fear responding. Both habituation and sensitization can be said to be forms of neuroplasticity, that is, changes in the nervous system in response to experience that essentially reflect that experience by way of learning. So whenever we see learning, we have to say, okay, there must have been some sort of change in the structure of the nervous system, and habituation and sensitization reflect that. Another form, a longer-term form of learning, is changing the actual structure of the synapse. So in response to repeated exposures over a long period of time, a synapse can either become habituated or sensitized by changing the number of connections that two neurons might have. So a habituated synapse might prune some of the axonal connections between one neuron and another. So it is actually chopping off some of its connections, and so the response of this neuron is going to have relatively less of an effect on this neuron over time. Alternatively, you might have a sensitization response in which this sensory neuron actually grows new connections And so the firing of this sensory neuron, so it sends its action potential, and that action potential branches into all of these different branches and will have an effect all up and down this cell. And so there will be a relatively increased response over time. So these will be somewhat longer term forms of learning because you're actually changing the structure of the cells, not just how they communicate, but also how much the communication is going to occur within the synapse.